So some symbols that are drawn from a distribution of English words, of sounds that we speak and so on, they need to be transmitted through a noisy channel, which could be, again, a wireless connection. It could be a wire, it could be a telephone wire, which it used to be at first. And that whatever is being put into that wire comes out corrupted as a Y by some sort of noise in transmission, limitations in transmission. And then the receiver receiving this output of a channel, which really is a physical thing, maybe if that's a cable, it's like a voltage fluctuation, needs to reconstruct back what was said, presumably with as little error as possible, right? So channel can be subject to various constraints on energy, noise, time, computational cost on the receiver end, and so on. There is a quantity underlying this schematic here that can be computed if you know this probability distribution. So the probability distribution of what you want to send, the probability distribution of, given that you are putting into the channel symbol what comes out, which has noise in it. So you can combine these distributions in a measure, a scalar measure, a mutual information measure in bits, which is a non-negative quantity that conceptually bounds or gives you a measure of the fidelity of transmission, how much information you can put through the channel without losing it due to these errors in transmission. Now, but the important thing for us is that Shannon already in his early work, you can read, I won't read, hinted that this theory is very generic, right? It talks about distributions. It doesn't talk about hardware, so it could be applied to many systems, not necessarily just engineered systems, also other systems. Later on, he wrote an essay called The Bandwagon, where he warned against too many scientific branches jumping on information theory and applying it all over the place without being very careful about how to do that, saying, I believe this will be applicable also in life science and biology and so on, but with a very careful measurement of what is meant by signal and what is meant by noise and so on. So was kind of a warning at that point. Belief in universality, but also warning about, being diligent. So what I would like to, the way I would like to motivate this talk is that if you now switch over from engineering to biology, the story of life and biology is actually full. It's replete with the concept of information. Okay, this word is used all over. And I'll start with the central dogma of molecular biology, right, that you probably all know, which is all about the transfer of information that's encoded in DNA to messenger RNAs to the structure of proteins, right? It is framed in terms of flow of information across these steps. And some of the work in our group, and actually I was yesterday giving a talk at Amolf in the group of Peter Reint and Wolle, is actually about these molecular systems that transfer and copy information, and how do we do that without, too many mistakes. In evolution, so Kimura is one of the fathers of theoretical population genetics. The word information, actually, just look at the dates, right? Was also very early invoked as saying, how much information gets accumulated in our genomes through the course of evolution, where, evolution selects for fit phenotypes and doesn't select and selects against non-fit phenotypes, and therefore kind of narrows down on, fit solutions, if you want, in sequence space. So how can you talk about that issue? In neuroscience, again, Horace Barlow is the father of what is called the efficient coding hypothesis. The hypothesis is that sensory neurons in your eyes and neurons in your ears have evolved to maximize the amount of information they transmit from natural signals into your brain, given the constraints that neurons face. Okay, let's say how quickly they can spike. Okay, and in developmental biology, Wolpert invoked this notion that's called positional information that underlies how multicellular organisms develop. The plan for today is I want to give you 3 stories, 3 vignettes, starting here, proceeding here, and ending here. All of them are, background plus some of our work, just to give you a sense of how these words that have been used in biology can be really formalized and developed into a more mathematical theory with which you can actually compute, as opposed to being just words and concepts. Okay, so I'll start with positional information in development. So let me introduce that topic first. And again, I'll stay around, so feel free to ask questions during or at the end if you want. There is many more details I cannot go into. So what happens during the development of a multicellular organism, you or the fruit fly? So here is the fruit fly, okay? So the fruit fly, the mother makes the egg. The egg starts as a single cell. Those cells then divide. This is sort of a, about 2.5 hours into development. It's a 0.5 millimeter sized egg with about 6,000 cells on the surface. They all look still the same, but there's some magic happens and you get this very clearly defined body plan, right, with lots of spatial structure that's reproducible from fly to fly. And so somehow what I'll try to explain to you is that the mom that makes this egg provides an input signal. The input signal breaks the symmetry, tells this will be a head, this will be a tail. So she deposits some chemicals here, but not here. That breaks the symmetry. And then in these cells, there is a complicated biochemical network, gene regulatory network that runs and creates a spatial pattern, very detailed spatial pattern, which will set the body plan and select this part will become the head, this other part will become the wings, this will become the tail, and so on. Okay. So there is another mode of development, which is actually for mouse and for us.
So there is one question from Valeria online. Why she doesn't see the slide online? Did you share the screen?
I think I did. I can. You are screen sharing, it says. So I can reshare the screen.
Yeah, reshare.
Participants can now see your application. Continue to share. Do you think they see now? I don't know.
I can ask.
So in the kind of development we have, this is self-organized in the sense that the mom doesn't provide any symmetry breaking signal, and yet still the cells self-organize and set up this body plan, which is also quite complicated. This is mouse development. And so what we wanted to establish in our group is an information theoretic way of thinking of how these spatially unstructured early steps end up in this reproducible generation of spatial structure. Okay, does it work?
They say yes.
Okay, they said yes, fine. Okay, so the key developmental biology question, actually it's a question that's very old in developmental biology, going back to this Wolpert's work, more than 50 years back, is the following. You start with a cell, that cell divides, all these cells have the same genetic material, right, the same DNA, and yet they need to still become different, and they need to become different in reproducible ways, right? They need to, what?
They say yes and again no.
Then I don't know what to do, sorry.
It says there also your screen sharing is paused.
Well,
I don't know why it's
you are screen sharing, and then when I do continue to share, and then it's paused. And then my mouse disappears, so I cannot click on anything.
You can press resume.
You have the desktop?
You can press resume, I assume.
I cannot, I do not have my mouse. I don't see my mouse pointer also disappears as soon as I do that. So I can, sorry for that, guys, but I can also try to just share the. I would like to be able to click resume, but then without seeing the mouse, I cannot click anything now.
Do you share your desktop? You just did.
I thought I did, right?
No.
Oh, the entire desktop. Okay, let me try that. Oh, sorry. Wait, out. And this. Okay. All right. So I hope it works now. So one of the key questions is, right, you have a lot of cells, they will have the same DNA, but how do they differentially be able to tell, I need to do something different than my neighbor? And, the answer is use a chemical GPS. Okay. So here is how we do it in engineering. If you want to know where you are, you have multiple satellites, the sources of information. You receive the signals from them and you triangulate your position. So you know you are here, and maybe that's why you should, do something or go somewhere. The concept of positional information, going back to Walpert, is a chemical version of that. In a chemical version, you don't have radio signals, you have chemical signals. So this is now space. This is a gradient of some molecule called the morphogen in space. Okay, and these are cells. Of course, that's a toy model. So if you are a cell, you can just locally measure the concentration of some chemical, right? And then based on this concentration, if it's higher than some threshold, you decide you need to become the red type. If it's, in the intermediate, you're white, and then it's blue, right? So you're doing a local measurement of concentration, and that is telling you, that's a chemical triangulation, right? That's telling you what you should become. Yeah.
So how stable is that the local information? Like, is this the static signal or is this like
It could be whatever. So this is just a conceptual model right now. I mean, this was proposed long time back. How could the cells do it? There was no claim yet that this is how they actually do it. This conceptual picture triggered experimental chase to find the first molecule that, if it exists, that actually does that, which is why Nobel Prize was awarded to Eric Wieschaus and Yanni Nussler and Volkart when they found bicoid as the first morphogen in the fruit fly development. I'll say some words about that. There are cases in some organisms where it's a static readout, so it's a more or less static gradient. You just read the local concentration, that's it. There are also cases where these things are very dynamic. Cells move and the gradients are changing, and that's a much more complicated thing. So let's say neural tube development in invertebrates. Right. So the conceptual proposal was there are special chemical gradients of these chemicals called morphogens that encode positional information. Mind you, that's just a word for now, okay? And this positional information somehow read out by cells locally to determine what they need to become. All right, so let's fast forward a few decades. How does this play out in the fruit fly? So this is a physicist sketch. Mom, lays down a fertilized egg. This is about 2 hours in development, I pointed out. There is about 60 rows of cells going along the long axis, about half a millimeter in size. If you wait for a bit longer, you would start seeing very reproducibly from fly to fly this morphological change where this almost single row of cells decides to invaginate and separate head from the rest, which is called the cephalic furrow. And what's really important is the position where this happens is extremely reproducible from fly to fly. What I mean by that, it's basically about 1% positional error. So 1%, the variation from embryo to embryo is about 1% of this whole length, right? 1% is roughly order of magnitude of the cellularization, right, resolution. So now, how do these cells know what to do? Okay, they all start out with the same DNA. If you zoom into at this stage before morphological change, what happens inside, and you do measurements of various gene expression across cells, and this is from the lab of our collaborator Thomas Greger, you will find that from head to tail, which is from head to tail, there are spatial, very elaborate spatial gradients of gene expression patterns in these cells. So here they are. They have names: hunchback, Kupel, giant, knurps. These are the famous gap genes, doesn't matter. They are extremely reproducible. So these envelopes here are variability that comes both from intrinsic noise in a single embryo plus across embryo variability. So typically about 10% variation from all sources. That's biological variation, not experimental noise. Okay. So there are these gradients here, and the suggestion was that these form the chemical GPS. So 4 gap genes form a code for where cells are. So I'll try now on the next slide to make this notion precise. Okay. So here is the quantifying the chemical gap gene GPS, and we really worked on this across many years now to try to kind of nail this down. So what's the basic idea? So here is just the same pattern replotted, head, tail. This is spatial coordinate, and these concentration gradients. The idea is the following. If you are a cell sitting here, 30% from the head in, let's say, right, and you measure a low value of Kruppel and Knurps and kind of intermediate value of giant and some value of hunchback, together, these four measurements are a code that you are at 0.3, like your GPS, right? Some other combination of concentrations corresponds to this position 0.6, etc. So I think our key suggestion at that time, as many years back, is that this concept that has been in the literature of biology of positional information, is just a concept, could be equated or identified with a mathematical quantity you can now compute from such data, which is Shannon's mutual information, which is really the information in bits that these four numbers carry about position. Okay, that's now a computable thing. And you might ask,
do you refer to multivariate mutual information or normal pairwise mutual information?
It's a multivariate. Okay. So this is to try to equate something from Wolpert with the sort of Shannon's mathematical formalism, and I'll get back to what you asked. So first question is like, why would this even be limited? Why would this number, be kind of limited? And of course, as an experimentalist, you have access to this type of data, which is a mean over many embryos plus the standard deviation over them. If you're a cell, in one embryo, you don't have access to that. You have access to four stochastic noisy numbers that vary from time to time, and they actually also are different from embryo to embryo because of all the sources of fluctuation. Because this is done at very small copy number of molecules. We are doing biochemistry with hundreds of molecules here. These are transcription factors. They're present at very low copy numbers. So fluctuations are large. This is thermal noise. It's embryo to embryo variability because when the mom deposits the signal, she doesn't do it precisely equally in every embryo and so on. So these sources of noise limit how much information these gradients have. If you do the estimate, you can first do the single variate if you want. Univariate, you can estimate how much information does an individual profile have about position. And you would find that all four of them typically care about 2 bits of information about position per gene. At that point, that was already a surprise because the textbook biology for these genes particularly, these were taken as a plain vanilla example of genes that define binary domains. This famous hunchback was said to be on here, sharp transition, off here. And if you have an on-off gene, the most information it can convey ever is 1 bit. It can split the embryo into front and back. That's the best it can do. If you estimate the information from data, it's 2 bits. What does that mean? It means that it's not only the plateaus that matter. These gradients, as sharp as they seem, actually contain distinguishable information. I'm not saying that the embryo uses it for now, but it's there. You can compute it from the data, okay? So each of these contain 2 bits. You might expect, since there are 4, that's 4 times 2, 8 bits. You would be wrong. These patterns are redundant, so these genes are not saying independent things. So if you compute the multivariate mutual information between all 4 of them together, which means you also have to be able to measure correlations between them, not just marginal values. So all 4 together carry about 4.3 bits of position information in this embryo. Now, what is this? Okay, so 4.3, is it a lot? Is it sufficient? Is it necessary? Can it be decoded? Question mark. Okay, how to interpret the number? So here I give you, it's just a short vignette of how to understand this number. So under assumptions of small Gaussian noise and so on, I will not go into the math. There is a very nice identity that equates the 4.3 bits, that's the information on the left-hand side here, with this non-trivial constants and this particular measure, which is called the positional error. So what is the positional error? So imagine you are a cell in the embryo and you measure these 4 noisy concentrations, and you now need to make a statistical estimate of where you are. And imagine that you have infinite compute and you can do base optimal statistical estimate. Okay, so meaning, given the known noise, you get you draws, right? Noisy draws, and you estimate your position. That position will have an uncertainty. This is the uncertainty. Okay, that's the positional error. This is the best you could do with a snapshot of 4 noisy numbers. Of course, that is now dependent on coordinates, so it can be different at different places in the embryo. And this equality equates the information in bits, which is a single number, with an average over space, that's just the embryo length, the average over space of this positional error. Okay, so 4.3 bits that we computed, if you use this formula, would correspond to a uniform, meaning equal in space, positional error of about 1%. And that is very nicely the rough order of magnitude of the nuclear spacing, because we have order 100 nuclei, 100 cells along the x-axis. Now, you will have noticed, if you're paying attention, that I said there is about 60 rows of nuclei. So from information theory, if I want really uniquely to specify each single row with 0 error, I would need log 2 of 60, which is more, which is 5.9, and I only have 4.3. And that's the difference between really 0 position error and enough information that every cellular row with 0 mistake says, I'm cell type A, I'm cell type B. And this 1% position error still permits 2 neighboring cells to have a mismatch probability because of the overlap of error distributions, right? So there is not perfect positional specification, but very good, okay? Now, is it used? So that's the biologically important question. We have this information, we can compute it, it's there in the fly. Does the fly use it? And for that, Julien Dubuis from Thomas's group measured the genes that are downstream of this chemical GPS. So one layer downstream from these 4 chemical, gap genes are the famous pair rule genes, which make also from head to tail. It's hard to see the staining in microscopy, but this is the analysis. So this is head, this is tail. These are the genes that make the famous in the fly 7 stripes, 7 troughs. And what you see here is each thin line is one embryo. This is not the mean, it's just one embryo to see that these are very reproducible, right? And you can quantify how precisely from embryo to embryo the peaks and the troughs are located, right? So this is plotted here. So this is head to tail. And this is now the precision, is the positional error of these peaks and troughs. It hovers around 1. So 1% is here. So along the axis of the embryo, things can be positioned reproducibly with 1% precision. And they're positioned by reading out the chemical GPS. Okay, so the chemical GPS has enough information to do it. And there is a downstream set of genes that actually does something with this information at the same resolution. Okay, what I'm not telling you is, because it's a longer explanation, but I'll give you the intuition, is not only that there is enough information, that this information gap genes is also, there is a signature, it's optimally organized. Why? It's because theoretically one can show that optimal information encoding in space predicts that positional information as a function of space is constant. It's not perfectly constant here, but it's constant. If you think about an engineer GPS system, if you're an engineer building the GPS, if you want a good GPS, what would that mean? It would mean you organize your satellites and your signals in such a way that no matter where on Earth you are, your positional error is roughly the same. You don't want to have places where your positional error is 3 millimeters and some other places where your positional error is 100 meters. Now, of course, there is Antarctica and Arctica, and the Earth is not all the same to us. But in a kind of idealized case, an optimal organization is such that no matter where you are, you can tell where you are with roughly the same precision, which is the flat positional error. Okay. Yes.
So, is there variability in the tails that would explain why we don't fit this?
So I'm not entirely sure, because in the actual fly, one thing happens that's not taken into account here, which is that the fly has very special patterning systems for the first and the last roughly 10-15% of the egg length, which are called the terminal patterning system. And so this is not part of this game. So it could be that the terminal patterning system takes care of the stuff in here. And it could be maybe more precise because you have this extra readout. What I really cannot show you because of the lack of time is that there is not just a proof of correlation. There is enough information and this information is used. There is also a whole set of work by Mariela Petkova and Thomas Gregor's lab where there is an evidence for causality, where you go into the fly and you mutate the maternal signals, you muck up with the chemical GPS, and you can quantitatively predict how all of these stripes will shift by how much, which ones will disappear, and so on. By just using wild type data, you can predict the mutants with no free-fitting parameters quantitatively. Okay, so there is enough information, the information is being used, and it's being causally used because you can perturb it and observe the consequences of that in this system. And in this system, it's all temporal snapshots. There is no dynamics needed to explain what's happening, but we think that's quite unique for the fly. All right, so now I switch gears a bit, and I'll now present still about the fly, but so I just told you there is empirical evidence that somehow evolution has really optimized the encoding and readout of this position information, data-driven. But now I'll try to do something else. I'll not start with data. I'll assume that position information is optimized, and I'll try to predict what happens in the fly. It's an optimization principle like in physics. I'll consider all sorts of gene regulatory models. I'll optimize position information in silico and then ask, does that optimal solution recapitulate what actually happens in biology? Can we have a proper, not fit, but a prediction? Okay. All right, so here is a short version. This was done by Tomek Sokolovsky, used to be a postdoc in our group. So what Tomek did is sort of a crazy model of multiple. So he modeled explicitly discrete cells along the long axis of the embryo, and within each cell there was a network of these 4 genes that could interact in any way, repress, self-activate, activate, whatever. In a parametrized way. So all these arrows can be parametrized in a proper dynamical, stochastic, spatial, dynamical model. So these are, think of this as, writing down a set of stochastic differential equations for the expression of these genes. These genes respond to the maternal signals, and you explicitly model all of that stuff on gene making mRNA, making the protein, degradation, the proteins can diffuse around, you put in the nuclear division. So it's a model I don't like per se, because we don't like working with 50 parameter mechanistic models. Okay, but it is a 50 parameter mechanistic model. The logic, however, will not be to fit it. The logic will be start with some random parameter values, forward simulate those patterns that you saw. Of course, there will be some crap. Compute the mutual information, the position information, and then adjust the parameters to maximize that position information, right? And try to find optimal solutions. So it's an optimization approach. Brute force numerics. It's terrible, okay? So you predict that. So what happens? So here is an optimization run. So you start with here is optimization steps. This is your objective function as position information we are optimizing. It's a variant of simulated annealing that, was done by hand. Start with some random patterns. The genes do something totally boring. Some are completely off, some are on, 0 bits of position information in this pattern. But as you optimize, you start creating structure. And at the end, you get an optimal solution, 4.2 bits of information. This is really, optimization in the computer. And this is your comparison pattern measured in the actual fruit fly. So claim is one of the information maximizing solutions is close to the measured pattern, and you can do this quantitative comparison. Okay, so you can, do R square and so on if you want. I think what's nice is that this is a fully mechanistic model that you optimize. So you can ask, how is it that gene expression can be so precise, which are questions developmental biologists are interested in. And the short answer here in this model is there is no single silver bullet. This precision really comes about through spatial averaging via diffusion, temporal averaging because of the time scales and how these genes cross interact. So it's not that there is one factor that, explains this. And, it has been an open question in developmental biology, how that works. What's nice is that what you optimize, this information is not just, log likelihood or chi-square. It's an actual phenotype that has a meaning, as I tried to convince you before, which you can independently estimate in the data, and there is nice agreement. And that's maybe a technical thing that I'll skip. I will say for all of you who are interested in optimization, this is a 50 parameter optimization problem. This is not the only solution. Okay, so there are multiple local degenerate solutions. Degenerate meaning they all reach about the same information. So it's not that this one is worse, but it's also not that it's better. So there is multiple roughly degenerate solutions. They are statistically similar to each other in the sense that they make all these slopes and peaks and so on, but how precisely those are positioned, that varies. For us, this is evolutionarily very interesting because this is Drosophila melanogaster. Okay, it's one fly species. This fly species has relatives. It is known that in those relatives, some of these patterns are flipped, they're changed, and so on, right? So clearly evolution, there is not just, one optimal pattern, and evolution itself has realized multiple. The only thing is that only for the Drosophila do we have these very precise measurements. For the other ones, we have qualitative observations, what happens when the domains flip and so on. So that's very interesting because evolutionary theory has this very long history of asking questions about historical contingency, necessity. Do you always get the same solution? Do you get different solutions? What's the role of selection versus developmental constraints and so on? Ivo Divo, tries to answer this question. In this model, what's interesting is that all of these questions play out, but in a model that is actually fitable to data, right? It's not a toy model. I mean, this is a mechanistic model of something that I would not be ashamed to show this as a fit, okay, with the mechanistic parameters and so on. But you can do now this in silico optimization, which is not evolution, but you can explore the limits that evolution is faced. What are the solutions? What are the trade-offs and so on?
So basically what you just said is that in this parameter space, 50 parameter space, you get it neutral evolution theory.
I'm not saying that. That's an extra statement. I'm saying there is multiple solutions that are degenerate. What I haven't said whether these solutions are connected or not. So is there a neutral path between these peaks? That is less clear. And I cannot answer this question because if you wanted to answer that question, are these peaks connected or are they separate, right? We would need to go down to the level not of parameters, which is what I optimize, but really genetic encoding into the DNA. And I don't have that link.
Instead of optimizing for the state, you can also optimizing for the trajectory.
Yeah, that would mean like, there will be an optimal that is just symmetric, like they had inverted.
Yeah. I mean, you could do that if you believe, but I'm trying as a physicist to say like, are the patterns that we see? I'm not making an evolutionary. I think I'm first making a statement. What we see in nature is actually derivable from the optimization principle. A separate question is how does evolution do that? I did it in silico, right, with the computer. I think the evolutionary questions are interesting. We want to go there, but there's still a few steps missing for those.
Because you have this paper on mutual information in the past space, right? The PRX, I think 2 years ago. And now I see that the mutual information multivariate is still on the state space. But if you generalize this into the trajectory space, you could also answer the question of whether. But
empirically, we know in this system, we know the snapshot, not from the optimization, from data, we know that the snapshot is enough. We don't here, right? It's not true in other systems. In the fly, somehow magically, a snapshot. Again, I'm not showing how we know that, but in the fly, a snapshot of gene expression at one point in time seems to encode everything that you need. So I'll be wrapping out with the first part. You can, of course, open the box now and look at the topology, right? So this is now, these are the 4 genes, and this is a biological consensus from the review of Yogi Jaeger of how they are supposed to interact. This is constructed, of course, by many, many students doing genetic mutants and so on, right? This is strong cross-repression here, weak side cross-repression self-activation. If we look at our optimal solution, you recapitulate this strong repression, most of the side weak repressions, and we also need some self-activation, although we don't get it in our optimal solution on exactly the same gene. So there is quite a lot of overlap. It's not exactly the same network, okay? Having said that, I think, I'm willing to say information maximizing solution almost correctly predicts the regulatory network topology ab initio. All right, so summary of this part. So I wanted to show you for developmental biology, you can take an old concept, which is position information, 70 years old, 60 years old, and you can turn it into a mechanism-independent physical quantity, information theoretic quantity that's now computable from data, which is the position information. You can try to elevate that into an actual function you can optimize, okay, in silico optimize to make first principles predictions. And I would venture out to say that, this type of prediction of a network, both topology, parameters, and patterns from first principles in this system is probably the first. So I don't know of anything in a way that matches the data. I already hinted that there is interesting implications or at least questions for evolutionary biology. And I'll just mention, if you're interested, there is a follow-up that extends this information theoretic formalism not just to positional information, but also to the self-organized systems that happen in us, Turing, patterning, lateral inhibition, cell, so there is a more general information framework to also think of the self-organized systems that is not instructed by the mom making the gradient. Okay.
By the way, for the optimization function, you only have the maximization, like normally, for instance, in the analogy with physics, you have the cost and benefit. So if I think about the benefit here, is this mutual information gain?
Yeah.
Is there some kind?
There is a cost. So there is a cost. Of course, I did not have the time to go into the detail, but the cost is very nice in these molecular systems. It simply comes from and that's an empirical constraint. It comes from the measured maximal rates at which you can make mRNA and protein. Right? So at which you can produce these molecules that form the gradients. So these maximal rates limit how many molecules at the maximum these cells can make. That limits how low the noise can get. Because if you can make more molecules, the noise will be lower. It's basically counting noise. And it's the noise that ultimately limits information. So if you're willing, so the cost is how many signaling molecules you're willing to allocate to make this GPS. Right? Basically, it's like building an engineering GPS. If you want to put more power into the transmitters and put more satellites, you probably can get higher precision. So in this case, more signaling molecules the cells can afford, the more precise patterns they can make, basically. So this is at the fixed molecular budget, which we know in the real fly. So we can solve the optimization with that fixed molecular budget.
And can I relate that cost for producing the signaling molecule to the metabolic cost?
No, I don't think you can. This is what we thought at first. We thought at first the cost is really in making molecules, and then every biologist told us that that's a stupid idea because the cells are making actin and myosin and all these cytoskeletal networks that are, orders of magnitude more costly than these signaling molecules. Why we do think it makes sense is because this cost that we put in, the number of molecules limitation, correlates totally tightly with something else. And that's time. It's time that we know. These genes are the fly, for reasons I do not know, evolutionary reasons, goes the amazing rate. Okay, this happens only within hours. Okay, and I think Bernard might have talked here about that. No? Okay, so he might have the answer for why it goes fast. Given that it goes fast, these genes here, and Thomas has measured this on the molecular level, so these genes are insane. So the cells divide, nuclei fall apart, then you cannot do anything, right? The nuclei reform, there is a break of 2 to 3 minutes before gene expression can restart, just because cells reform and morphogen flows back in. Polymerases lock onto the gene, and then essentially you have polymerases driving on the gene with maximal speed and ribosomes on the transcript with maximal speed. So this thing is expressing the fastest rate it goes. And I think, saying that you have a fixed budget of molecules translates in this setup of in this time, you can just make that many, not because in ATP metabolic costs they're expensive, but because molecular machinery can just not make more. Okay, so I think that's the constraint. It's the time constraint that this needs to go fast. All right, so Good. When did we start?
I think 5 past 11.
Oh, okay, fine. I'll have time for one more story. Okay, maybe I skipped the evolution.
But it can go.
I'll ask you. I don't want to go over time, so I'll finish on time. Ask if whoever wants to leave, and then I can I can go on. Yeah. So I'll now completely switch gears, just erase, right, tabula rasa, and switch to neuroscience. Okay, and give you a story which I hope is interesting from the efficient coding hypothesis in neuroscience. Again, it's an information story. And it's this is a very old, I would say this is one of the oldest applications of information theory to a biological system that has a history. So, no, we are not the first to do this at all, right? So there is a venerable history, and the thinking goes as follows. Why is that? Is because it's also the closest to Shannon's conception, right? Think about your eye, that's the retina. Your eye keeps getting stimuli, complicated ones, natural stimuli with all the richness and the unfolding time. It goes into your photoreceptors and local processing in the retina. And what the retina does, right? It encodes everything you see in the stream of neural spikes. Okay, so there is a bunch of axons in your optic nerve that are conveying binary spikes to your brain, and this is where all your visual information comes from, right? I mean, if you cut that, you're blind, no? And then, of course, the brain does something with these spikes. Guy uses them to make decisions, blah blah blah. So this is almost a bona fide application of information theory, right? There is stimuli that come with some statistics from the outside. There is an encoding device right here, which is biological. It's not engineered, but it's biological. And then there is, a stream of symbols that get sent, and the brain decodes them or whatever it does, right? And Barlow early on formulated this hypothesis, which is not in words, which he already thought of as a quantitative hypothesis, as like sensory systems, for instance, the retina here, invest their limited resources. So one has to say what that is. What is the limited resource? In order to maximize information transmission from natural stimuli into spikes. And then, whatever the brain does with them. But this bit is a transmission channel. The optic nerve is an information transmission channel. So, of course, now all these underlined words are things that we need to make precise, okay? Information can be made precise by information theory, okay? And so this is Shannon. So you can say this is really an mutual information, or the information rate, if you want, because it unfolds in time, between stimuli and spikes. That's a thing that's computable. Maybe not easily, but one can compute that, right? Right, so you can compute it, and this is again optimization theory, right? It says they maximize this information, right? So somehow, whatever the retina evolution has pushed the retina to do whatever it does in a way that maximizes this transmission, given constraints. Constraints, what do we think of constraints? For this system, the constraint is typically, this is metabolic constraint in this case. It is a constraint on neural spiking. Neurons can only spike at certain maximum rate, order 100 hertz, and spikes really cost. They're really metabolically expensive. I don't know if you know, but per gram, retina is the most expensive tissue in your body. It doesn't weigh many grams, but it's very expensive. If you're deprived of oxygen, your vision first goes black because that tissue doesn't get enough energy, okay? And then you lose consciousness and so on, right? So it's a very expensive, transmission device, okay? So why would this theory make sense? And it makes sense because it's a theory about encoding of natural signals. Natural signals have a lot of statistical structure. But here is an image. I don't know, I'm making it up. That's about megabyte in size, because in like naive encoding, you encode 3 colors for every pixel, right? And that is roughly what your photoreceptors do. They are kind of a pixelated array that encode the light intensity with 3 different color channels at every point in space. However, that nearby pixels in natural scenes are strongly correlated. If that pixel is white, a good guess is the next pixel is white. And so if you're doing compression, if you're doing efficient coding, you don't want to say white, 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 white. No, I mean that because that's the default. You just say white, same, and here is a change. Okay, that can be compressed down to use the redundancy in the signal. You can compress almost without the loss, and you know that this is actually what compression algorithms do in your computer, right? But your neurons can do the same thing, and you can compress this down to a much more compact representation, which is thought that low-level vision, retinal ganglion cells, and primary visual cortex does that. Okay, that's the thinking. And then, of course, there is even higher level descriptions, right? This is not just a pixel, right? There is like high level objects like trees and so on. So you can describe this, at the more and more compressed level of representation. And the thinking is that actually this is our visual stream in the brain, right? Going from photoreceptors, LGN, retinal ganglion cells, primary visual cortex, and then higher order visual cortices. So meaning stimuli that we want to encode, they're redundant. There is a lot of statistical structure. There's something that's called sparse. I don't have time to explain what this is. And they're predictable in time, meaning there's time correlation. If I look at this picture now, a moment later, it's almost the same picture, right? It's not a new random image, right? So there's correlations across time. This means those are compressible, okay? However, neurons, and unlike your digital hardware, neurons and signals are noisy, and so you cannot totally compress out everything. You need some error correction capability because neurons sometimes spike stochastically, right? So relying on a single spike would be a mistake, okay? And you would see a lot of flickering in your image if you did that because your neurons sometimes spontaneously fire. And then the third bit of theory that you need is okay, maybe not all bits are equally important for the brain. So the brain might prioritize some bits and not other bits, right? So there might be different importance, and that meaning there is semantics to that. And so I'll not go into that, just to say that these objectives can actually be folded into a unified information framework, theoretic framework of efficient coding that says like, these are the stimuli, how should they be encoded? Can we predict something about how let's say our eyes work? So here are now for you some historical successes of that theory, not from us, these are old. So maybe the winning, the holy grail was the following prediction, that if you look at retinal ganglion cells in your retina, so neurons in your retina or other retinas, right, they will perform filtering of an image, like convolutional filter of an image. And the convolutional filter, one can predict this via this efficient coding. One can say, like, what kind of filtering should a neuron do to optimize information transmission? And you would predict this type of structure, which is called the center surround. The neurons, this is space. The neurons sum up luminance levels in a small radius and subtract away, an annulus, luminance in an annulus. This makes sense because if you have very, very highly correlated images, on a uniform white background, this filter gives 0, right? It integrates the center, but the center is the same as surround, so it just subtracts and doesn't report anything. This is a change detector, right? It detects, only responds to spots, okay, not to uniform. This is like in your convolutional neural nets. Now, if you do, kind of machine vision, you'd have convolutional filters. What kind of filters are they? Well, some of them look like that, okay? So information theory predicted that this is the filter shape. This is the measured filter of one of the neurons in the visual stream. This is the center, that's the surround. This has been replicated gazillion times, okay, across animals, in the retina, in the LGN, and so on. So this center surround structure can really be derived ab initio as optimal, and neurons do that. There was some very nice other stories I'll just mention because it's fun. Just good to know. You all have your trichromats, right? You have cones that respond to 3 colors that are roughly red, green, and blue. Not exactly, but roughly red, green, and blue. And you might wonder, if you go to a healthy vision human and count how many of red, green, and blue you have in your eyes, you will not find 30%, one third, one third, one third. You find about 8% blue, and the rest is allocated between red and green with huge variability. okay? Almost tenfold variability for healthy color vision people, so not colorblind. And the question is, can this be predicted from first principles? These numbers, okay, now 8%, 92, and so on. And there is a very nice series of papers where you take a color calibrated camera, which really means, a camera that you calibrate to photon fluxes in various wavelength, windows. Can go to savanna, which is thought to be our primordial habitat. You get the natural image statistics, and then you solve the following theoretical problem. I give you n neurons. You can apportion them between 3 color classes that we know the spectral sensitivity of. How should you apportion them so that they will encode maximal information from the natural scenes you measure? And you get the numbers right. You get 8% of blue. You get even more nicely objective function. The information is very sharply picked at the blue, so that you should have. But for the red and green, it's actually quite flat in a large plateau, which is where human variability sits amongst individuals. Still perfectly healthy color vision, because it doesn't actually affect information transmission so long as you have some and some of one and some of the other in a pretty broad range. Okay, and then it drops, so you cannot be without one or the other. Then you're colorblind. Okay. So that's a very nice set of results that is in this field. The other nice observation, as a colloquium style, is that you can make an actual information rate estimate for the information that goes through your optic nerve. Okay, we know how many cells there are. We can estimate the information rate through each single retinal ganglion cell. We can sum this up. It's about, for guinea pig, it's about 1 megabit per second. Guinea pigs see well. For humans, it's probably a bit, it's a bit more. But I find this personally very impressive. There is a biological cable that is transmitting at essentially 1 megabyte per second for humans, okay? You might be then tempted to think that if we do Netflix video, where your whoever is focusing on the video, right? If you ask for video with high quality, what's the data stream that the computer needs so that that it delivers high quality? If you think carefully, it should be the same, right? Because if your eye is compressing the data stream very well, and my computer is compressing the image very well, of course, if it delivers the image at 100 megabits per second, it's wasted, because that's not what can go through my optic nerve. Okay, if it delivers it at 10 times less, then I'll see pixelated bad resolution. Okay, so that's interesting. And it's also interesting that despite the fact that your optic nerve is a very expensive tissue, you can think how long it took us to develop electronic devices that can actually play back movie at that data rate without consuming a lot of power. I mean, we can do it now, but it's relatively recent, right? I mean, so it's a pretty good, data processing device. So that's the retina. So here is another prediction. These are neurons in your visual cortex, or this is mouse probably. They also have filters, but their filters that they act on the image are not center surround. They are these Gabor-like elongated filters. This is also something that you would do in image processing as edge detectors, right? These are little edge detectors. They're band pass, they're oriented, they're localized, and so on. These can be also predicted from efficient coding theory. Okay, so and this is about higher order statistical structure in natural scenes. This is about pairwise correlations natural scenes. This is higher order, higher than pairwise order. The natural scenes contain edges and corners. These are not pairwise features, and this is what your cortex is extracting. And then the last, this is really cool. This is from auditory periphery. So what you see here are little waveforms. These are filters of the neurons in the auditory nerve. And you see 2 colors. You see blue and you see red. The red one are prediction of efficient coding by recording natural sounds and ask how should, if you have a filter bank, what kind of filters should you have to optimally encode natural sounds? And in blue are actual measurements from the nerves. They lie on top of each other. I mean, this is quite spectacular. And also there are biological evolved systems for which you have an ab initio prediction that is really, quite amazingly good if you're coming from natural science. All right. So Okay, so here is our contribution to this. This is like, can we go beyond filters? That was the question. Can we go beyond how neurons in the sensory periphery filter stuff to something that's higher order cognition, like perception? And this was in collaboration with Jonathan Victor. There was an amazing observation, a hypothesis actually known as a Euler's conjecture about which textures humans can perceive and which visually can discriminate from random or discriminate from each other and which not. This is just to give you an illustration how it looks like. So that's a texture. It's a synthetic texture, okay? And if you don't, human observers don't need much time to figure out that in this square, things are somehow different than elsewhere because you have little pluses in here and you don't have little pluses there, okay? What's harder to recognize is actually also stuff in this square is different. But for that, you need more time. So in this square, there's little t's. Here are corners, here are t's, and here are crosses. Crosses from this, that's very fast. This from that is not that fast, okay? There have been a ton of psychophysics experiments done on this human perception. Usually they are done in such a way that you flash such an image for a very short amount of time, let's say 200 milliseconds, and then humans have to tell in which part of the image, either here, 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 or here, so it's usually four-fold choice task, was the texture different? Do they see it, right? And then you measure, is it above chance or not, okay? Now, Euler was making in 1962 a conjecture that says humans can only differentiate textures if at the level of the pixels, those textures differ at the pairwise correlation order in luminance, spatial pairwise correlation, right? So if you measure spatial pairwise correlation function here and here, if it's different, you will see the difference. If it's not different, you won't see the difference. So that was a hypothesis, okay? So we are sensitive, hypothesis was we are sensitive to pairwise spatial correlation in natural scenes. Now that hypothesis was very quickly turned on its head by a counterexample. So here is the counterexample. It's called the isodipole texture. So this texture, it's not random black and white checkerboard. This looks more like random black and white. Every pixel is just an independent 50% black and white flip. That's certainly not. But what you can show is this is a particular mathematically generated texture that has the following property. Every block of 2 by 2 pixels you choose has no bias in luminance. There is equal number of black and white. There's no pairwise correlation of any kind in this image. There's no third order correlation of any kind in space, but there is fourth order correlation. Fourth order in the sense that every 2 by 2 block of pixels is parity. It has exactly even number of black or white, not odd, never odd, okay? So this is a fourth order correlation that you clearly see is different from random. Now, what I'm not showing you is that if you generate textures where you put the same fourth order correlation between 4 pixels that are not arranged in the square, but are arranged in these other Tetris shapes, which are called gliders in this field, so like this or like that, you would see something that's totally non-random. You could test people on it, flash it for 200 milliseconds. You don't need conscious scanning by the eye. You can, it's called a pop-out, right? You see it and it's here. It's not random, okay? So if you put fourth order correlation between pixels arranged in these shapes, you see it. And the surprise comes if you put the same amount mathematical of fourth order correlation between pixels arranged in these shapes, you see this, which in a 200 millisecond presentation, you do not discriminate from random, okay? So question here was, and this is, kind of perplexing, our brain obviously has neural mechanisms, actual compute mechanisms that can detect some type of fourth order correlation, but not some other type of fourth order correlation. It's a really qualitative difference. It's two groups. It's not continuous. You see this ones quickly, you don't see those ones. So the question was why? Is there an explanation? And there was like decades of work trying to find it's about the symmetry of these things, it's about, whatever, the surface ratio, and there's always a counterexample. There is nothing obvious. So what we thought with Jonathan Victor is that maybe this is not about this synthetic stuff. Maybe it's about natural scenes, okay? In terms of efficient coding, what our brain has evolved to detect and see is related to what's out there in nature. So what we decided to do is to take the natural scenes and do some very big injustice to them to process them in some way, which I cannot go through. So we take samples from natural scenes, and in natural scenes, we can extract the presence of, if I take 4 pixels like this, I can extract the presence of bias, black or white. That's first order marginal. I can extract all pairwise correlations, vertical, horizontal, diagonal. I can extract third order correlation and this fourth order correlation, right? I can get this from data using what is known as information decomposition, second order information that's statistical structure present in pairwise, in triplets, in quadruplets. There is some technique to do that. And so what I can do in natural images now is ask, if I take 4 pixels, is there non-trivial statistical structure present on the fourth order correlation in natural scenes? Okay. And I can, do this as following, right? I can ask, let's say, just to introduce, I can take, let's say, to start simple, I can take 3 pixels and I can measure, is there third order non-trivial structure present between the 3 that cannot be predicted from knowing all pairs? And for this particular case, you would conclude, no, this is not predictable in natural scene from knowing all pairs. There is something non-trivial at the third order, right? There is something more on the third order, maybe relevant for us. For the square glider, 2x2 glider, is this fourth order structure predictable if I know all the triplets or, all the pairs? And the answer is no, is not predictable either. However, if I take this other shape, is the fourth order correlation between pixels arranged in this shape predictable by knowing all triplets? The answer is yes, that is. There is actually nothing new if we look at pixels arranged in this way in natural scenes. So that's a property of natural scenes. It's not a mathematical necessity by any means, okay? And so what's interesting is that for all shapes where the answer is no, those are precisely the group 1 gliders, those that if I make a synthetic texture, you will differentiate from random. For all shapes where in natural scenes there is nothing new that's present, if I generate synthetic texture with fourth order correlation, those are group 2 gliders, which is fantastic. They have the fourth order correlation and we're just simply blind to them. Why? Because in natural scenes, you don't need to detect that. There is nothing in natural scenes that's interesting on that order. And so our brain simply did not evolve mechanisms to extract that.
Could have a conclusion that the best measure to measure correlation in the visual cortex is a 3-point information like total correlation, or?
Well, this is not the information in the cortex. That's the information in the images, right?
The image.
Yes, yes.
But if the efficient coding holds, you would just...
Well, very good question. I mean, then I'll comment on this. Yes, where is this represented in the brain? Okay. So if you want to view our visual cortex as a GPU, I mean, the fact that you can tell that it's a pop out 200 milliseconds, it means that our brain really has parallelized processors that simultaneously look at the entire scene and extract that. So it's not cognitive conscious processing where you scan consciously the image. Okay. So the brain has invested into this parallelized hardware that extracts only those pieces of information that are actually informative about natural scenes. Now, in the synthetic world, we can isolate and only make a texture that has only that fourth order correlation that's never in natural scenes, and then you don't see it, right? Okay. So that's the qualitative statement. And then we wondered, can we make a quantitative, not a yes, no, can you see, can you not, can you actually make a quantitative prediction? And for that, Jonathan Victor really tortured more human observers. You will see the psychophysics experiment is really a torture. So now you focus on 4 pixels arranged in a square, and it's kind of nice to look at, no? You can generate these textures. So these are now mathematically fully defined. They're maximum entropy textures, actually, for those of you who are interested, where you can modulate first order. You start here, it's random. You go to just white bias, black bias. That's just probability of the pixel being black or white. You can induce pairwise vertical correlations a lot or anti-correlations. Horizontal, and you can see, right? Vertical structure, horizontal. You can do diagonal pairwise. You can do triplet correlation, so you get these triangular structures emerging, and you can do our quadruplet fourth order that you have already seen. So you have seen fourth order here, right? These weird checkerboards that happen, or anti-fourth order. And now you can display, you can now continuously dial these knobs and ask at what point the humans start differentiating the texture from random. Okay, that's called perceptual sensitivity. So you can map across humans how sensitive we are to dialing in or out these texture components. So that you can do in the lab. And then in the image analysis, what we have done is we went to natural images and we measured the variation in natural image across these texture coordinates. You can think of these as texture coordinates. They parameterize texture in some mathematically well-defined way. So how do these things vary and co-vary in natural scenes? Right? And then we wanted to ask in terms of efficient coding, if we know what happens in natural scenes, we should be able to predict how sensitive humans are to these various manipulations, right? Right, and so here are some of the results. So these are perceptual sensitivities to changing pairwise correlation, vertical, horizontal, meaning higher number means we are more sensitive to seeing that. So if you put a little bit of that texture in, you'll see the difference from random. Very sensitive to horizontal and vertical pairwise correlation, a little bit less to pairwise diagonal. We're actually least sensitive to third order correlations and slightly more to fourth order, which is interesting. It's kind of an inverted order. These are different human subjects. It's quite reproducible because some of them are new, they have never done this task, and some of them have tens of thousands of hours of torture time on this psychophysics. It doesn't change much, actually. You go to natural scenes, and the natural scenes will predict this same order. These are different versions of the analysis, just to show that it's quite robust, of how much variance is present along these coordinates, texture coordinates, correlates with how sensitive we are. There is still one free-fitting parameter to match these things, which is just to match the scales of these things, because they're different. One is perceptual sensitivity, one is variance in natural scenes. So it's not that striking, it's okay. However, here is now the torture. So, this order you predict correctly, but now the torture comes where Jonathan Victor started not just varying one of these coordinates, but two at the same time. So you can you can create textures that have a mixture of, let's say, pairwise diagonal and, let's find it, pairwise diagonal and third order triangular. And so now, of course, you can, you're dialing two knobs and you're saying, how much do I have to displace, how much admixture of both do I have to see a difference from random? And of course, now you don't get perceptual sensitivity, you get the isocontour of perception, which is an ellipse, right? How far away do you have to go from the center to start seeing, this is different from random? So first of all, what you see, so here is human data, here are human elliptical perceptual sensitivities. Each individual is an ellipse, and this is really reproducible across human observers, no? Here is the individuals, here is their mean, okay? Now you can ask about can this be predicted from covariation in natural scenes of these texture coordinates? And I think this is the impressive bit, okay? So there is no longer any free-fitting parameter here. The dark circles are predictions of efficient coding from natural scene covariation, and the reds are averages across subjects. So, and there is no clear, we try to rationalize this and so on. There's just no obvious order. This is a property of natural scenes, right? That these ellipses come out the way they do. So, maybe I summarize this bit. Efficient coding in neuroscience has really quantitatively predicted extremely well certain non-trivial properties of sensory neurons in your vision, in your audition, ab initio, by optimizing information flow from natural scenes, and really important, from natural scenes, right? The predictions would be totally different if we evolved to look at random noise. We do not get center surround if you look at random noise. You only get it if it's efficiently encoding natural scenes, right? We try to push it in our group to go beyond sensory periphery into more central processing, right? These perceptual sensitivities and so on, they don't happen in your eye. Actually, there is follow-up work to show on what we have done, to show that this perceptual sensitivity to texture emerges after the primary visual cortex in what's known as V2, the secondary visual cortex. This has been generalized not to binary textures, but to grayscale. And actually, very recent work in Trieste has trained rats to discriminate these same textures as humans. Rats have the same order of perceptual sensitivity as humans in natural scenes. So they see fourth order better than third order, as we do and as predicted by efficient coding. You can train them by giving them a reward if they can discriminate. All right. So in neuroscience, efficient coding as information maximization has really predicted well the central brain processing across different species. And to summarize it, this really means nervous systems commit limited resources to optimize bits per unit cost for encoding natural signals. That's what efficient coding is. And maybe I finish now because I'm over time. And so if you want to take some questions, I still had like 5 slides, but maybe I just put conclusions on, no? And then I'm happy to discuss. So I think this kind of story of biology is really full of the concept of information, but there are cases, I'm not saying it happens every time, that this concept can really become a cornerstone for a quantitative and predictive theory. That's quantitatively predictive, you can predict stuff ab initio, and it's really like an interdisciplinary field, between biology, evolution, physics, information theory, and so on. So it's quite interesting. Maybe I would like to, for those of you who don't know, I'd really like to, and are thinking about biology, I'd really like to advocate an old work of David Mars from computational neuroscience that's called, he introduced the 3 levels of analysis. And recently with David, we took it from computational neuroscience, giving credit, of course, where it's due, and started thinking about developmental systems in that sense, which is really an advocacy for thinking about all of these systems on 3 different levels that are not competing. There are just 3 different levels of description. The mechanistic level, of course, what are the molecules, processes, networks, neurons, cables, whatever. Then there is an algorithmic level, right, which says what kind of computation is this biological hardware making. And then there is a normative level, which is what I was talking about today, which is, is there a theory that predicts why this computation should happen, right? Or in other words, what would be the optimal solution to a task? What is the algorithm that approximates the solution? And then how can this algorithm be realized in hardware? So in neuroscience, this has been quite influential. But outside of neuroscience, I realized people don't actually know that much about these David 3 Mars levels. And oftentimes, let's say in my biology, colleagues still argue about, is it correct to describe systems in the mechanistic model or this other model? And it's not about correct, incorrect. No, I mean, these are alternative views that, are complementary, not correct, incorrect. And so, our starting point really has been, can optimization of information flow become one of the, of course, normative theories for biology? And it's simply a restatement of this, which now everyone knows with deep learning, right? That compute is costly. I mean, compute is costly also for the brain. It's costly for the organisms. And what I did not show you on evolution is that evolution can also be recast as accumulating information into our genomes. There's certain ways to encode information in the genome and certain other ways. And what I would have shown you had I had enough time was that evolution long-term will prefer ways to encode information in the genome where less information needs to be maintained in the genome to realize the same phenotype that you need, right? So there is some sort of a version of how to do compact representation in the genome that's quite predictive, and you can use it to make predictions. And then the last bit, which is methodological for those of you who want to make contact with data, these are all optimization theories. Optimization theories in multiple dimensions, let's say like the fly, are very notoriously hard to compare rigorously to data. How do you know the theory is correct? It has multiple solutions, has multiple parameters. Those parameters are not independently measured. So what's the statistical test, rigorous statistical test, to be able to show this theory works? What does it mean works, right? I've shown you receptive fields predicted and measured. I rely on your brain to look at 2 pictures and say, oh, it really works. The 2 pictures are the same, but that's not a statistically rigorous comparison, okay? So we have taken some time with Victor Mlynarski on this to really come up with a proper Bayesian way of testing high-dimensional degenerate optimization theories. We haven't solved the problem. I think it's baby steps, no? But I think that's also important. How do we test these things? All right, so maybe I stop here, and then if somebody wants to hang around and chat, I'm happy to do so.
Have some questions?
Yeah, coming back to this data correlation question, because it's buzzing if you not neuroscientists, I didn't know about this, that you can perceive order dependence better than three order.
Mm-hmm.
If you translate to encoding like a network or interactions within the brain, what's the best function to encode that has this property?
So what I can tell you is the following. So if you and that's one of these follow-up experimental works by Jonathan Victor. If you go and record from cells in the primary visual cortex, while you are showing these weird textures, the random noise and the fourth order, you will not find individual cell responses that can discriminate between these two textures. So the information is there because you can do it, we can do it, but it's not on the single neuron level in primary visual cortex. You have to go one level higher in the visual V2, secondary visual cortex, and there you can find an individual neuron, which obviously is pooling information from multiple neurons in the primary visual cortex and somehow combining it probably in some nonlinear way. There you can find an individual neuron which will have responses that discriminate between, right? So there, the information about fourth order correlation in natural scenes gets localized into individual neuron responses, right? So then you don't need any longer to have some coordinated representation with many neurons. Single neurons will do it, but only as high enough. So you have to go through several of these linear and nonlinear processing layers, right? So in primary visual cortex doesn't happen. So I think it's, in terms of kind of today's deep learning, right? I mean, you need to go through a certain number of these nonlinear stages so that you find representations that are, where unique neurons represent these higher order spatial correlations in natural scenes. But I think what was also nice is from the neuroscience side, nobody actually knew how to. So for primary visual cortex, we know experimentally what kind of stimuli we have to display to the animals to excite V1 neurons. You display gabors, you display little edges, and the neurons go crazy. That's another, that's kind of the famous V1 oriented filter contribution. For V2, for the next layer, nobody really knew what you need to show in an experiment to make those neurons really like the stimulus and respond. You could show natural scenes, and they would, of course, respond. But in natural scenes, it's hard to like what is responsible in natural scene, right? It's a superposition of a ton of things. So it's actually, in terms of synthetic images, these textures are kind of the first clean mathematically statable ensemble of stimuli that you can generate, and it's mathematically well defined, that will differentially drive the V2, this kind of next layer, right? So it has this value as a probe, okay, as well, right? And you can make them more complicated because you can combine these texture components and so on. Yeah, they're in Maxent in the sense they are the most random. It's a distribution of textures of these colorings that is most random while constraining the statistics that you specify, right? That's how they're generated. Yeah.
The question. I think we only have another question.
Yeah, sure. Thank you.